Welcome back to Dead Reckoning, the Human Life Expectancy channel. In this video, we'll focus on how genetic engineering is being applied to plants, animals, and also to humans, giving us unprecedented power to enhance human life expectancy and accelerate volitional evolution. Let's start with plants. As we discussed in our video, Is Evolution Evolving? Over the last 23,000 years, human selective breeding has resulted in the dramatic, beneficial, and volitional evolution of almost all domesticated crops. You may recall that in the late 1920s, the pace of evolution quickened when scientists discovered that we could increase the rate of genetic mutations by bombarding DNA with chemicals, radiation, and enzymes in a process called mutation breeding. This caused extensive and unknown changes to DNA that mostly resulted in unsuccessful, unviable organisms. However, some inevitably proved viable and more to humanity's liking. From an evolutionary perspective, it gave breeders of plants and animals a broader spectrum of mutations from which to choose, accelerating volitional evolution. Between 1930 and now, well over 3,200 so-called mutagenic plant varieties contributed to the Green Revolution in the 1950s and 60s, and resulting in common varieties of grains and crops that we eat today. In the 1990s, we developed the ability to make targeted changes to DNA using genetic engineering, resulting in genetically modified organisms, or GMOs. This was a big step forward because instead of creating random unintended mutations through mutation breeding that caused both good and bad changes throughout a seed's genome, targeted changes could be achieved to specific areas of DNA, greatly accelerating the volitional evolution of plants. More recently, the genetic engineering tool called CRISPR has been used to improve food crops. It's produced new, more resilient, faster-growing, and more nutritious wheat, corn, rice, and other crops, and can protect them from disease and inclement weather and save millions of human lives. For example, vitamin A deficiency is estimated to kill 670,000 children a year under the age of 5, and cause an additional 500,000 cases of irreversible childhood blindness. To address this problem, genetic engineers produced biofortified golden rice with biosynthesized beta-carotene, which is a precursor to vitamin A, in the edible parts of the rice. The regulatory agencies in the United States, Australia, New Zealand, Canada, and the Philippines have all approved the use of this rice. CRISPR is also being used to produce a strain of rice that's able to grow in salt water, which could be critical as climate change causes oceans to rise, flooding low-lying rice fields. CRISPR is also used to make other grains and starches more robust. Wheat with higher fiber and lower gluten content, waxy corn for better cornstarch, potatoes and sweet potatoes that are disease and bruise resistant. Tomatoes and fruits such as apples and papayas are made more disease and heat resistant. Also, CRISPR may be necessary to save oranges and bananas. The citrus greening disease caused by the bacterium Liberobacterium asiaticum is a serious threat to orange production. It's characterized by streaks of different shades on the leaves and deformed, poorly colored, and unsavory fruit. In areas where the disease is endemic, citrus trees live only for 5 to 8 years and never bear suitable fruit. The disease was discovered in Florida in 1998 and has now attacked nearly all the trees in that state. It was also reported in Brazil in 2004 and has been spreading rapidly. Fortunately, the University of Florida has just been awarded $4.5 million to develop a CRISPR-based cure for this important disease. Bananas could also be saved by CRISPR. Before 1950, a type of banana called the Gros Michel was the main variety produced. But they were attacked by a fungus called the Panama disease, which wiped them out. It was found that a slightly less tasty form of banana called the Cavendish could grow in soils infected with the virus. They quickly replaced the Gros Michel and have been used as the global standard ever since. However, in 2008, a mutated form of the Panama disease began to attack the Cavendish as well. CRISPR-based genetic engineering might be able to help the Cavendish to become immune to this disease and save this important crop. Genetic engineering is also increasingly being applied to animals. In a prior video, we showed that over the last 15,000 years, domesticated animals, including dogs, chickens, pigs, cows, oxen, horse, and many others, have evolved dramatically from humans' volitional selective breeding. Recent advances in genetic engineering have greatly accelerated the pace of this evolution. In 1972, Paul Berg, who later won the Nobel Prize in Chemistry, created the first recombinant DNA molecule when he combined DNA from a monkey virus with DNA from the lambda virus. Then, in 1974, Rudolf Janisch at MIT created a transgenic mouse by introducing foreign DNA into its embryo and giving us much greater control over the volitional evolution of animals. 8 years later, transgenic mice were developed that passed the transgene to their offspring. In 1984, mice were genetically modified so that they were predisposed to develop cancer. The first transgenic livestock were produced in 1985, and the first animal to synthesize transgenic proteins in their milk were mice, engineered to produce human tissue plasminogen activator in 1987. In December 2003, glowfish, a zebrafish with a fluorescent gene added to allow it to glow in the dark under ultraviolet light, became the first genetically modified animal to be sold in the United States. The first genetically modified animal to be approved for food use was the Aqua Advantage salmon in 2015. The salmon were transformed with a growth hormone regulating gene from a Pacific Chinook salmon and a promoter from an ocean pout, enabling it to grow year-round instead of only during the spring and summer. Today, animals are genetically engineered for 6 basic purposes. The first purpose is biomedical research. Genetically altered mice, also known as transgenic mice, and other small mammals can be used as models in scientific research to represent other mammals and test potential cures. By knocking out genes responsible for certain conditions, it's possible to create animal model organisms of human diseases. Mice, rats, pigs, and other animals are genetically modified to carry a specific gene of interest. So for example, based on Rudolf Janisch's discovery, mice are now commonly engineered to increase their susceptibility to cancer, making them useful for studying cancer and its cures. They're commonly used for in vivo models in cancer research. Animal models have also been used to study obesity, heart disease, diabetes, arthritis, substance abuse, anxiety, aging, and Parkinson's disease. Research has also proceeded to expand the capabilities of genetic engineering. Researchers at UC Berkeley used CRISPR to generate a large number of bodily transformations in crustaceans. Gills growing where they shouldn't be, claws becoming legs, jaws turning into antennae, and swimming limbs becoming walking limbs. In 2021, a team of scientists and entrepreneurs announced that they've started a new company named Colossal to genetically resurrect the woolly mammoth. The company aims to place thousands of these magnificent beasts back on the Siberian tundra thousands of years after they went extinct. Genetic engineers are using synthetic biology to write new genetic code that nature never imagined to make organisms do things they weren't previously programmed to do. One recent study showed that we could engineer mice to have all female or all male litters with 100% efficiency. More to our point, a 2016 study by cell biologist Darren Baker and Jan van Deursen showed that genetic engineering can increase the healthy lifespans of mice by 25%. Much more on this and other methods of enhancing life expectancy in future videos. The second purpose of genetic engineering in animals is environmental monitoring. For example, in 2000, M.J. Carvan showed that genetically engineered fish could be used to help monitor pollution in underwater ecosystems. The response of certain enzyme systems to organic chemicals, such as oil hydrocarbons, PCBs, and dioxins, makes analysis of enzyme levels a potent way to monitor pollution. Third, animals are being genetically engineered for protein production. In our last video on genetic engineering, we showed that yeast and other animals have been re-engineered to produce proteins that humans and other animals lack. Mass quantities can be manufactured by growing the transformed organisms in bioreactor equipment using industrial fermentation, and then purifying the protein. Some genes don't work well in bacteria, so yeast and cells of other animals are often used. These techniques are now used to produce medicines such as insulin, human growth hormones, follicle-stimulating hormones for treating infertility, human albumin, monoclonal antibodies, anti-hemophiliac factors, vaccines, and many, many others. Together, these save millions of lives a year. Fourth, animals are being genetically engineered to help find cures for human diseases. In addition to serving as models in biomedical research, as mentioned above, genetically modified mice are used to develop drugs and cure diseases. Transgenic mice are used for drug development as they facilitate target validation and the study of response, resistance, toxicity, and pharmacodynamics. They can also be used for introducing disease resistance in genetically modified breeds. As another example, researchers are altering the genome of pigs to induce the growth of organs with human DNA to be used in transplants. Fifth, animals are being genetically engineered to manufacture products. Genetically modified organisms now produce a range of products useful in industry. For example, genetically modified bacteria are now being used in bioremediation, where the bacteria are used to convert pollutants such as crude oil, carbon, plastic, and other toxic waste into less toxic forms. Modified bacteria are also used as sensors that express fluorescent protein under certain environmental conditions, and genetically modified algae are used to produce biofuels. Other uses include using bovine collagen to make non-animal leather, manufacturing yeast with spider DNA to make ultralight silk stronger than steel, and using renewable microbes to produce acrylics in paints. The sixth purpose of genetically engineered animals is for increased food quality. Livestock animals have been engineered to grow faster, be healthier, and resist diseases. Examples include cows that grow without horns, which can cause injuries to farmers and other animals, are resistant to parasites and heat, and even produce allergy-free and human breast milk equivalents. Pigs that grow larger, more quickly, and combat diseases. In December 2020, the U.S. Food and Drug Administration approved genetically engineered pigs for use in food products. Pigs can now be used to produce meat that's safe to eat for people with meat allergies. Pigs have also been modified to be immune from disease and have less environmental impact. Scientists are in the process of developing CRISPR-altered piglets that are immune from porcine reproductive and respiratory syndrome, PRRS, which causes an astonishing $2.5 billion of losses annually. Genetic engineers have also created the EnviroPig, a line of Yorkshire pigs in Canada that excrete 30 to 70% less phosphorus in their manure. The lower concentrations of phosphorus in the surface runoff reduces algal growth because phosphorus is the limiting nutrient for algae. Because algae consume large amounts of oxygen, excessive growth can result in dead zones for fish. Also, goats have been engineered to produce milk that fights off diarrhea in humans and even has strong spiderweb-like silk proteins. Chickens have been engineered to grow larger, faster, and to lay many more eggs. There's also genetic work underway to develop hypoallergenic chicken eggs. And as we mentioned before, Aqua Advantage salmon were genetically engineered to grow year-round instead of only during spring and summer, and found to be safe for human consumption by the FDA in November 2015 and approved for the U.S. market in 2019. The successful, safe genetic engineering of plants and animals has now led to genetic engineering of humans. Genetic engineering has many applications to medicine that include the manufacture of drugs and gene therapy. Let's start with drugs. As we saw with COVID-19, genetic engineering has allowed us to quickly manufacture mRNA vaccines that save millions of lives. Under normal circumstances, making a vaccine can take up to 15 years. Prior to COVID, the fastest vaccine development was 4 years for the mumps. Thanks to advances in genetic sequencing, researchers successfully uncovered the viral sequence of COVID in January 2020, roughly 10 days after the first reported cases in Wuhan, China. They were then able to share the sequence with scientists around the world who developed the basic architecture for the vaccine in about 48 hours, using a cutting-edge approach for developing vaccines called mRNA, for messenger ribonucleic acid. Traditional vaccines put a weakened or inactivated germ into our bodies to trigger an immune response inside our bodies. That immune response, which produces antibodies, is what protects us from getting infected if the real virus enters our bodies. Instead, mRNA vaccines teach our cells how to make a protein, or even just a piece of a protein, that triggers an immune response. The vaccine shuttles the mRNA into cells which read those instructions and churn out the protein that triggers an immune response inside our bodies. Genetically engineered viruses confer immunity without causing infections. This process has allowed scientists to move with record speed. Less than 10 weeks after the Chinese scientists published the COVID sequence, a team from the National Institute of Allergy and Infectious Diseases and the biotech company Moderna had a candidate ready for phase 1 trial. The Moderna vaccine was built with mRNA, the piece of genetic code, using the coronavirus's spike protein. The Pfizer-BioNTech COVID-19 vaccine was the first mRNA vaccine to be used in humans outside of clinical trials. The unprecedented speed with which COVID-19 was sequenced and a vaccine was developed proves that vaccines can now be produced in a fraction of the time they used to require. COVID has resulted in millions of dollars of investment in this mRNA technology, and the dramatic success of these COVID vaccines has advanced the technology to the point where many believe that we may be close to universal vaccines that will protect against a broad range of conditions, including flus, HIV, cancer, emerging diseases, and even pathogens, malaria, and other parasitic conditions, with a large potential impact on HLE. In addition, Kaojian Mao and team at Shanxi Medical University showed that genetic engineering could bring us drugs that could dramatically reduce heart disease. A number of single bases that are related to heart disease can be knocked out in a single course of treatment of genetic engineering delivered by a lipid nanoparticle. This delivers a large protective benefit from lifetime low LDL cholesterol levels with no adverse side effects. Also, in 2021, Wai Zhao and colleagues at the University of Minnesota showed that it was possible to regulate weight gain by modulating the activity of the inflammatory cells. This could become a blockbuster drug that could significantly impact global HLE. Plenty of other genes can be knocked out using drugs. Here's a list of just some of the over 10,000 diseases that can be cured by knocking out single nucleotide pairs. And in addition, there's over 40,000 diseases that involve more than one gene that could be knocked out. Let's now move on to gene therapy. We're at the very beginning of applying gene therapy to humans. In short, classic CRISPR technology is not yet precise enough for many therapeutic applications. Also, currently about half of disease-causing mutations in the human genome can't be targeted by CRISPR. However, a 2019 study by Andrew Anzalone and team found that prime editing approaches, including cytosine and adenine-based editors, CBE and ABE respectively, offer the prospect of broadening this to almost 90%. Gene therapies can now be used to enhance human life expectancy in multiple ways. In addition to the reduction in genetic diseases resulting from IVF and PGD, as we discussed in our video on revolutionizing human reproduction, genetic engineering of somatic or body cells could cure many human diseases, extending HLE. If genetic engineering repairs the genome and restores the production of healthy proteins before it causes irreparable damage, then a one-time treatment should provide a lifelong cure. CRISPR pioneer Jennifer Doudna says, quote, "In virtually all cases where a particular mutation or defective DNA sequence can be linked to a pathology, CRISPR can in principle reverse the mutation or replace the damaged gene with a healthy sequence." This includes conditions such as cancer, HIV, genetic disorders, dwarfism, Alzheimer's disease, high cholesterol, diabetes, fertility, and even obesity. She writes, quote, "It's becoming difficult to find diseases for which CRISPR hasn't been mentioned as a possible therapy." Great progress is being made in using genetic engineering to treat cancer. Cancer is caused by inherited and acquired mutations in DNA, creating an enormous opportunity for genetic engineering to cure or prevent cancer altogether. CRISPR can be used to treat heart disease. A study published in 2021 by Kiran Musunuru and team showed that in vivo CRISPR-based editing durably lowers cholesterol in primates. CRISPR was used to lower LDL cholesterol in monkeys by 60% in just 1 week. And the monkey's cholesterol levels stayed low for over 8 months after a single dose of treatment. This has the potential to dramatically reduce human heart disease. In respiratory disease, modifications to the BMPR2 gene can help treat pulmonary hypertension. Also, Jennifer Doudna recently founded a company that uses CRISPR as a tool to change the genetic instructions that cause disease. The company, called Scribe Therapeutics, recently announced a collaboration with Biogen to develop CRISPR-based genetic medicines for neurological diseases, including ALS or Lou Gehrig's disease. Genetic engineering could also help in our battle against obesity. Mice have now been engineered to be completely resistant to diet-induced weight gains. Since obesity drives heart disease, cancer, diabetes, and many other of the primary causes of death, this offers the prospect of greatly reducing mortality rates and increasing HLE going forward. More broadly, genetic engineering will likely allow us to treat or cure many diseases, and perhaps even aging itself, resulting in a significant extension in HLE in the next 20 years. However, uncertainty remains regarding exactly when we'll have this capability and when regulations will allow its broad use. We'll cover these points in future videos. Accordingly, though, we use several scenarios for the rates of development and adoption that led to our dead reckoning that HLE will expand by at least 10 years in the next 20 years. We'll continue to watch developments of this revolutionary technology closely and keep you up to date in future videos. We'll also address what makes us think that curing aging might actually be possible, how it might be done, and what regulatory issues will need to be addressed. In the meantime, please hit the like button below, subscribe to the Human Life Expectancy channel to make sure you don't miss our next videos, and post comments for the topics you'd like to have us cover. Thanks for joining us. Till next time, as we say at Human Life Expectancy Inc., hi hua! Please, do what you can to help yourself and help us all to live longer.